Hello everyone, welcome to this extra session for problem solving for NPTEL Animal Physiology course. And in this session, because it's an extra problem solving session, because this session is beyond our regular 12 weeks session where we are discussing each week the topic that was discussed in that week, and then we go ahead with like set of questions that cover those topics. Today, this is for a revision live class. So it's an overall revision for the entire course. So what my plan is that I will just take you over this course and whatever we have discussed so far in the 12 weeks. This is like a summary of the entire course. And what you would find here is that we will look over all the topics that we have done in the 12 weeks and how are they connected, and some important topics I will just go over them once again. And at the very end I have set of questions which are for all the 12 weeks courses, 12-week lessons. And if we have time, I will also go over those questions one by one. Otherwise they will be there at the end of this video, and you can always attempt on them on your own. Yeah. Okay, so let me begin. So this is the summary of these course and the problem solving sessions that we have had. So the course Animal Physiology has had 12 weeks. It's a 12 weeks course, and each week had 5 lectures. So overall for this course you have a 60 lectures syllabus. And for the problem solving session, we have had 12 problem solving session. The last one will be the on the 12th problem solving session, which will be after today's session on Monday. And 12 plus 1, this is the extra session where we are discussing overall what we have looked into this course. And if you would have followed my different videos, you would have seen that we have covered the topics in the lectures using several modes of questions. For example, we did MCQs, we have done fill in the blanks, we have done true and false, we also did match the followings, and we have done assertion reason. So all of these are like they're distributed across the 12 weeks like videos that you will see. And they are covering most of the topics that were discussed during that week, right? So, because you are already at the end of this session, let me begin with what we started with and how did we move ahead in each of the week that was there. This will be a long video, around 2 hours, and the whole agenda is that this can be a review, or yeah, it can be a revision for you of what all you did in this course. So, if you remember, or this is how your course is structured. You have 12 weeks. The first week, if you remember, we started with the introduction to anatomy and physiology. Then in that week, after you have an idea of what is animal anatomy and physiology, we moved ahead to organization of living systems. So how are they organized? So if you remember, the muscles, the tissues, the skin, we discussed about these. We also had section where we looked into homeostasis and how it is and about the system integration. So basically, how is the balance maintained in the body, and what is the positive feedback loop in hemostasis. So basically we were looking into how your body is regulated, how there is a equilibrium or a balance in the body with regards to its chemicals, with regards to how it functions, and how is that regulated. It means that if there is any kind of deformity or if any kind of perturbation in the system, the body attempts to bring it back to normalcy, right? So that's about your hemostasis. And the regulation and feedback, positive and negative, were about how is this regulated at different scales, right? And the next one about chemical basis of organization of the body. So where we were looking into different kind of atoms, molecules, cells, tissues, all the biomolecules that your cell is constituted of, your proteins, your DNA, your RNA, how are they present, what are their roles, what are the kind of biomolecules that are present in your cell. So this part covered that. And once we had a brief overview of how our body is structured and what are the components of body, we started with the different physiological system of our body and we discussed them in great detail. We started with skeletal system, if you remember. So we in skeletal system we started with integumental system, if you remember, about skin, nail, hair. After that we moved on to bones and cartilage, and we discussed about how bones are formed, how what is cartilage, what is the difference between bones and cartilage, right? And then we moved to muscles, right? Muscular system. So we understood what are muscles, what are the different type of muscles, and then we in depth we looked into skeletal muscle, and we because skeletal muscle, we know that it is what is driving your movement, right? So we looked into the anatomy of skeletal muscle. We looked into the contraction of muscle, how does muscle contract, and then we did something called sliding filament theory. We discussed about sarcomere. And in relation to this we discussed about actin and myosin, right? Now we did about skeletal system, we did about muscle, then we started an entire uh, section in this middle of the course to neural system, where we were discussing almost everything about neurons and how does the neurons propagate the information and the action potential and the signal transduction, right? So if you see here, so in neural system, as you can see, there were 4 weeks of course material for neural system. We started by understanding how does this function and what is the connection, and then we moved on to the problems associated with neural system in case it stops functioning, or over the period what happens to neural system and how does those autoimmune neuronal disorders these come into place. So we first for so the how the flow is that you start with skeletal muscle. So let me just take it quick. You start with the entire skeletal system. Then from there you move to skeletal muscle. Now we know that skeletal muscle, we are moving the skeletal muscle. We are moving our muscles. How we are moving? Because we are governed by some signals. How are those signals related? Their signals are related by neural system. But before understanding the connection between these two, we had to first understand the neural system as a whole. So we started by understanding what is the length and tension relationship, what is about excitation and contraction coupling. So we will understand here contraction. So how does this contraction is coupled with the excitation of your neuronal cells or cells which can propagate signals, right? And then we understood about the stretch reflex phenomena, which also was discussed later in further slides, right? We looked into the nervous system and understood how the signaling takes place. We understood what is neuron, how are they structured, how are they connected, and then we looked into the ions. So basically we looked into their excitability. What is this excitation term? How do they propagate the signal? What are the ions that conduct the signal across? So, and then we understood the importance of neurotransmitters, action potentials. Okay. And then we moved to epilepsy. So, all the associated disorders, we moved to epilepsy, long term potentiation depression, Alzheimer's, Parkinson's, ALS, spinal cord, and then glial cells, just as a topic which is part of your neural system, but or which is also included in the neural system, right? So we covered all of these, and then, like I said, we discussed stretch reflex arc again, and then moved to other systems. So we did about skeletal system, we did about neural system, and then we have these special senses, which are basically you can think of them in terms of sensory organs, right? So you read it about hearing system, which is about ears, olfaction, which is about taste, anatomy of eyes, or a whole week about eye visual system, and then we had heart, followed by lungs, and then followed by the last week, which was discussing about the remaining system, for example, endocrine system, digestive, kidney, blood. We have already done the previous week also, and reproductive system. So you can see how we started like all the systems are covered in this course, and each of them are covered in a way that you understand the working of the system, and you can then understand if something is not functioning properly, what can it disturb and how can it affect your body. Okay, right. So I will just walk you through now all the subtopics that we have done in the videos. Now, these will look repetitive to you. So the whole agenda of this extra session is to give you an overview of what we did in this course. So maybe you are more comfortable with the first few set of lectures, but not with the second half of the course. So you can skip that video and go to that part and try to see these different topics. But my suggestion is you can watch these video in bits and parts also. In each of these, I will just give you the link, connection link and the important topics that we have done and describe those important topics once again, just for your reference. Okay, so right, let's begin. So we started with living organisms. We described living organism with these characteristics. The organism should be able to respire, should be sensitive, so its sense organs should be functioning, should be should be eating, so you have nutrition and then excretion, then reproduction. They should also be growing and they should be moving. Now, if you see, these all terms are basically governed under different physiological system. So muscular system, respiratory system, all these special senses that we talked about, digestive system, excretion system, reproductive system. So all of these systems are what you have discussed in this whole course. Yeah. So now the course says it's animal physiology. To understand what's physiology, physiology is basically study of how living organisms function and how they maintain life. So all this system, how together they function and maintain the life, maintain how we are surviving. That's physiology. And it explores how the mechanisms are going on, which are basically letting us live. So all the nervous system, musculoskeletal system, circulatory system, respiratory system, everything is in function even now when I'm talking to you. So how is this connected and how are they functioning so that I can sit here and talk to you and not have problems associated to these different systems, right? So the functioning, if you understand the functioning, then you can understand any disturbance to the functioning. So to treat or to understand the problems, you need to understand the basis first, and that's what we are going to do, right? So this part, okay, in every such videos, I will keep giving you an idea about the week number so that you have the idea of which reference week I'm talking about. What I mean is, let's say I speak that this is part of week 1, which basically means that if you want to see the topics in detail, or if you want to have more problems like in the sense MCQs or other questions relating to this topic, you can go to the channel and go to problem solving week 1. Yeah. Right, so we already discussed about physiology, right? So let's begin. Just a second. So I spoke about the way the body or the organism is structured, and this is what I meant by it. So we have these different level of organization. Any living organism is organized in these different levels. This, you can go to the minutest level, which is your chemical level. So your chemical level have atoms which come up together to form DNA. These DNA form up together as to make a cell. Those cell can be different types of cell, but those different types of cell come together to form your tissue. The tissues form organs. Different organs coming together forms your systems, and these systems together form your organism as a whole. Again, let me just break it down to you. Let's see here, you have smooth muscles. So you know what are the different type of muscles: skeletal muscle, cardiac muscle. So all these muscles take cells, make up the tissue. So let's say smooth muscle cells make up smooth muscle tissue. Skeletal cells make up skeletal tissue. Okay. Now, smooth muscles tissues together make up an organ. Let's say the stomach. Okay, now the stomach is an organ. You have similarly have other organ like small intestine, large intestine, liver. All these different organs together make up digestive system. And then all the systems, for example, digestive system, here you will have your cardiovascular system, here you will have your respiratory system, muscular system, all these systems together make up your organism level, right? Okay. So let's go to the first, I mean, we have, I already told you in chemical level, we are talking about DNA, RNA, protein. From there, we move on to cell, which I also told you, smooth, skeletal, and cardiac. So that those uh, together come up to form the tissue. Now, tissues are again of three different types. You have epithelium tissue, you have connective tissue, you have nervous tissue, and muscle tissue. So these basically tell you where they are present. Epithelium is anything in the covering. So you have cuboidal epithelium, you can have simple squamous epithelium, you can have stratified epithelium. Then you have muscle tissue, which are present in all these muscles, the smooth, striated cardiac muscles. You have connective tissue. You have tissues which are present in between two tissues. And then you have nervous tissue, which is surrounding the nervous cells or neurons. Okay. Then we looked about the integumentary system, if you remember, and we started with skin. So you can remember that we looked into the structure of skin. Basically, I am talking about, so this is your skin. What are the layers of your skin? So you can see that we have dermis. What we see here right now is epidermis. We started with epidermis, which have these hair shafts, which you know already. Below epidermis, you have dermis. And then if you remember, in your dermis, you had several layers. In the dermis, you had several capillaries, touch receptors, hair follicles, which the base of hair follicles, which come up as hair shafts. You have these sweat glands, sebaceous glands basically here. So the ducts are there. And below dermis, you have the subcutaneous layer, right? If you remember. Now, why is this important? Because skin, because of the presence of all of these, have several functions. Functions like sensation, which is because of these receptors, for example, touch receptor here. Heat regulation, because you the sweat produces sweat. Sweat gland basically produces sweat, which is allowing for your thermoregulation, so heat regulation. You have absorption, so you remember you can the cells here can absorb. Uh, the skin can also absorb from the application. They're protecting the underlying material. They have excretion, sebum, sweat, right? And then they also have vitamin D protection. So there are multiple functions of skins, and that's why it's important for us to know the structure of skin. Now, if I already told you about the different layers, just looking at this once again, you have the epidermis, and this is your hair follicle. Like I said, this is hair shaft. So the one that protrudes out is hair shaft, but the one that is at the base is called the hair follicle. Together they make up hair. Then you have the, so let me bring you here. You have the dermis, right? So dermis have three layers on their own. You have dermal papillae, papillary layer, and reticular layer. And then you have subcutaneous layer. Okay. But if you remember, just a second. In the epidermis also, I don't know if you have seen that first session or not. Sorry, week 2. So second week session. Epidermis itself has several layers. It has stratum, basal, spinosum, granulosum, lucidum, corneum in order. Right. So if you remember, we discussed that these cells will be basically arranged in layers. So you need to also remember the different layers in the epidermis structure. Epidermis is a part of skin. Epidermis itself has 5 different layers. So you need to remember those different stratas, right? Similar to what dermis. Dermis has these 3 different strata. So you also need to remember these 3 different stratas. Okay. So in skin, it is important that you remember which first of all, which layer we are talking about, and let's say epidermis. And in epidermis, you should remember the different strata of the epidermis. Yeah. Okay. Next, if you remember, we discussed about nail and hair anatomy. So basically, what I mean here is that so along with skin, you also have nails and hair, which is again part of your integumentary system. So we had seen what are the structure of the nails, what is present around the nail, we know where is nail, we have the nail plate, we have the matrix, we have the root. So how do they grow? And similarly, we looked into hair anatomy. I told you we already have discussed this. We have the hair shaft, we have the hair root, we have the hair follicle, melanocyte, which gives it the dark coloration. And even in the hair shaft, you have three layers: medulla, which is a central layer, which is covered by cortex, and then which is covered by cuticle. And to all the people who take care of hair, you will know that we always talk about the cuticular hair and keeping it soft and keeping it having no bristles. So whenever we talk about the type of hair, like it's very frizzy or it's very smooth, we're generally talking about cuticle. We want to keep it smooth, but we also talk about medulla, we talk about hair follicles a lot. So that's why understanding the anatomy of these simple structures becomes important for understanding. Okay, so after your integumentary system, which covered skin and your nails and hair, we moved to bones and cartilage. And if you remember, so what is cartilage? Cartilage is present here, the soft tissue. And we remember there are three types of cartilage. You have elastic cartilage, you have hyaline cartilage, and you have fibrous cartilage. What is elastic cartilage? It's basically it's present in your external ear, as you can see here. This one, this is elastic cartilage. So it has basically it has chondrocytes and elastic fibers in the matrix. Okay, so if you see cartilage, it's basically a layer of cells, and you have these fibers and you have these chondrocytes, which are the cells of cartilage. Okay, so in your ears, in your epiglottis, in your auditory tubes, you have elastic cartilage. Okay, then you have hyaline cartilages, which again have chondrocytes in their lacuna, but these chondrocytes are all packed to each other. Those are stacked chondrocytes, and then the matrix. And these kind of stacked chondrocytes can be found in your synovial joints, so in between the knees. You can find them between the tips of your rib and the sternum. So wherever you have, these fine boundaries between your internal structure, that's where you have this hyaline cartilage, for example, nasal septum, even trachea and bronchi. So all these finer uh, lining is where you have the hyaline cartilage. And then lastly, you have the fibrous cartilage, which are basically, as the name suggests, these are collagen fibers in the matrix. So these are basically found in structure which requires strength. So for example, in knee joints, the padding in the knee joints, or in the intervertebral discs, so which requires your strength. So these are the cartilage which are found in different regions. Now for bones, if you remember, we discussed about two different kind of bones. Let me just first take you there. So we discussed about two different kind of bone. We had discussed about spongy bones and we had discussed about compact bones, right? What was spongy bone? Spongy bone is also called trabecular bone. If you remember, these are basically found in long bones, and so your compact bone is covered by a spongy bone. So what is compact bone? These are basically heavy bone. They're tough bones. So they're basically the tight structure, and around them you have the spongy fabrics, right? Spongy tissues. So these are the spongy bones, and they are covering the compact bone, right? And bones have their own energy reserves. If you remember, we discussed about yellow marrow, we discussed about red marrow. So yellow marrow was basically anything which is yellow in color is basically fats. So yellow marrow in your bone is basically the stored energy for the bones. They're storing lipids. Red bone marrow is basically blood cells. So they are responsible for hematopoiesis, formation of blood cells basically. So you will have bone marrow where basically bones itself has its own reserves and stores for uh, blood cell generation. Okay, so that was about bone and cartilage, and then we also looked into different kinds of formation of bones, right? We basically looked into many different kinds. So first of all, on this slide here, you have endochordal ossification or endochordal bone formation, right? So you have formation of a bone collar around hyaline. We discussed about hyaline cartilage. So what does it say? That wherever there is hyaline cartilage, that's the place where you have this endochondral ossification. So you have the bone formation around those cartilage. the first thing that happens is cavitation of the hyaline cartilage. So the first thing is there is a cavity formation. Now, in that cavity formation, you have invasion of internal cavities by the periosteal bud and spongiform formation. So the first thing that will happen here is your invasion by a bud, which is basically what will give rise to your bone, and then begins the spongy bone formation. As after a few time, you will then have a medullary cavity as the bones keep forming. So basically, you have appearance of secondary ossification center, for example, epiphysis. Okay, so here you will see. So it starts from here and keeps growing, forming different other centers, which again takes up the job of elongating the process. And then you have these epiphyses, which come up, which basically, whenever they are complete, so hyaline cartilage remains only in those regions and everything else is bone, right? So endochondra is when you have it from the hyaline cartilage. And the other one was intramembranous ossification, which is basically condensation of your mesenchymal cells into soft sheet permeated with blood capillaries. And then those osteoid tissues, they are deposited by osteoblast. So you have the osteoblast. So what are the difference? Chondrocytes and osteoblasts. Chondrocytes are present in cartilage. So that is by cartilage. These are by osteoblasts, which are basically responsible for bone cells. One has involvement of chondrocytes and one has involvement of osteoblasts. This is how you need to remember. One is present intramembrane, one is present around the hyaline cartilage, right? Similarly, and for cartilage also, if you remember, you had interstitial growth and you had appositional growth. So these are the words that you need to remember with respect to your bones and cartilage. So, and we have discussed all of these in the respective problem solving session. So do go back to those respective videos, and these are discussed in more detail there. Okay. Right. Now we have done the integumentary system, and we have looked into your skins, your hair, your nail, and about your bones and cartilage. Now let's move to muscle. So we start with the cardiac muscle, skeletal muscle, and smooth muscle differentiation. And I told you they are where they are present. We know that cardiac, as the name suggests, is present in heart. Skeletal in your muscles, which are involved in movement, and your or rather they are surrounding your bones. And smooth muscles are the ones which are present in your organ system, so for example digestive system, right? Now you can differentiate all these different muscles based on their appearance. So you have striated is basically presence of these dark and light bands. So if they are striated, they are, and they are involuntary. Involuntary is basically moving those muscles is not under our control, right? So involuntary and striated are cardiac, striated and voluntary are skeletal. Involuntary muscles which are non-striated are smooth. Very simple to understand. If they are non-striated, they are smooth in appearance, so that's why they are smooth. And digestive muscles are not under control, so they can remember it as involuntary. And it's easy when you remember to distinguish it like this, that striated involuntary is cardiac muscle, striated voluntary is skeletal muscle, right? Okay. So then we had this extensive discussion on skeletal muscle and its function. So we started by first looking into the structure or the anatomy of skeletal muscle, right? So if you look here, you have, this is how the muscle here, let me just show you. This is a muscle, how it looks, right? So you start by calling it a muscle layer, but when you keep checking the cross section, you will see how it is structured. So the first thing is, if you see the cross section, you will have the covering called epimysium, and you have these bundles here. All of these bundles are basically fascicles. Now each of the fascicles, if you see the cross section, you will see that they consist of several myofibrils, right? Now all these myofibrils are basically stacked, so that there are multiple myofibrils. In each myofibril, if you bring the cross section, you will see that they have the sarcolemma and a sarcoplasm. Again, you have these several fibers here, right? These coverings here. These are, each of these is a muscle fiber. So your muscle fiber, if you take it further, you will see that it is basically consisting of actin and myosin. right? So basically all the actin myosin are packed in a muscle fiber. Multiple muscle fibers are packed in a sarcolemma, sarcomere, and all the sarcomere together make up a myofibril. Myofibril together make up vesicle. Vesicle make up your muscle, right? Okay. So whatever we just discussed about, let me just go back and tell actin myosin. We will see it now how they are present. Okay. So this is your myofibril. Let me just show you once again myofibril. These are myofibril. So one of these strands is what I'm showing you right now. Okay. And in this myofibril, let me just walk you over the structure first, and then I will tell you what happens to these different lengths. Okay. So see here, these lines which are present here, they are called the Z line. Okay. Then you have, as you can see, you have these interspersed here, one line running here and one line running here. So these are the blue ones are the thin filament. Thin filaments are the actin, right? And the red filaments that you see here, these are the thick filaments. These are called the myosin. So you have myosin, actin, myosin, actin, myosin, actin, right? So across a Z line, it basically between two Z lines, you have your sarcomere. So this is your Z line. So between two Z line, you have sarcomere. Okay. Now, if you look each of the sarcomere, look into this length here. This is how a sarcomere looks. You have actin, myosin, actin, myosin, actin, myosin. You have a myosin in between two actin filaments. And the central line here is M line. Okay. Now, when you, there are also banding pattern to it, which is called A band and I band. So if you see the A band here, it's basically this. A band is basically your myosin thick filament stretch. So you can see here. I band is the region without any myosin, just the actin. Okay. So what you need to remember is this basic idea. What happens to this basically when this muscle is contracting, that becomes easier if you remember what is the particular region referring to. What I mean is, we all know what happens when contraction takes place. Your uh, A bands goes, if you can see here, without me telling you, A bands become smaller, or rather I bands become smaller, right? This you need not categorically remember. If you remember what is exactly happening when sarcomere is contracting. But if you remember I band constitute actin and actin filaments come closer, you automatically know that when your sarcomere is contracting, I band will get smaller. So this you might not want to remember, or you might not need to remember. But you need to remember the individual patterns. So you need to remember Z line, M line, A band, I band, thick filament, thin filament. Okay, this you need to remember. Now, what is this sarcomere trying to tell you? So basically, like I just told you, you have all these different I band, Z band, A band. Now, when there is sliding filament theory, or whenever the sliding filament theory takes place, your actin basically slide over myosin. So your thin filaments slide over the myosin filaments, and the sarcomere shortens. So contraction is shortening, right? So your sarcomere will shorten. When your sarcomere shortens, the Z lines, which are at these boundaries, they come closer. As they come closer, your so A band, which is your myosin, doesn't change. So A band stays the same length, but your I band, which is your actin band, gets smaller, and the H zone, which is basically the central part of A band, where only, so here, this zone is H zone. Okay, this is disappeared. Now, to understand it better, this again we have done in the problem solving session, but just for your understanding here, when these actin and myosin interact here, these pull them closer. This H zone, which is now only of myosin fragments, filament, is H zone is disappearing because you have all these actin coming over each other, right? So these come here, which means these are also getting pulled, and these are also getting pulled. So all this distance becomes very small. All this distance goes disappearing. But what is same, this length does not change. Means basically your myosin doesn't change. It just pulls the actin closer, right? So that's your sarcomere, and that's all about your sarcomere contraction. And how does that take place? That is governed by the sliding filament theory. So what does sliding filament theory basically tell you? Sliding filament theory is basically telling you that how your muscle contraction is taking place uh, to produce movement at this microscopic level, which will be then shown as a muscle movement later, right? So what you see here is, this is the first thing that takes place. We already know this is a thick filament, right? So your myosin, it cross bridges to the actin on the actin myofilament, and during this, because it already has its ADP and PI, inorganic phosphate, with it, so this is called stroke. So this working stroke is basically the myosin. So you see it is connected like this in a straight position. What happens next is the head pivots. So the head moves like this of the myosin, it moves like this and it bends. And as it does that, it pulls the actin filament sliding over the M line, right? So that is what is sliding. So it is pulling it, it is sliding over a M line. Then what happens by utilizing ADP, and as the ATP attaches to the myosin head, the crossbridge detaches. So as the movement has taken place, ATP will come and bind, and it will then go back to its original position. It has to relax again, right? It can't stay in contracted position. It has to relax again. So the contraction is what will give the movement of the muscle, and the muscle will then go back to its original position due to ATP hydrolysis. Okay, so the ATP energy is utilized for your muscle movement, is what the whole sliding filament theory talks about. Okay, and then with relation to this, you have tropomyosin and troponin and titin. All of these also you should remember. These are all proteins which have specified role in the movement of muscles. So you should remember these as well when you are looking into sliding filament theory. Okay. Then a very interesting topic, which is length and tension relationship, which basically talks about how does your length change according to your contraction, but at the same time also tells you what is the optimum desired length, right? So basically, length and tension together will determine how the force is generated by this muscle fiber, right? So, and what is this length that we are talking about? The length is basically the distance between the Z line. So let me show you again. Z lines and Z lines, this is the length, right? So this is the sarcomere length we are talking about. So when you're talking about sarcomere length, you're basically talking about these myosin and the two actin filaments that are coming across, right? And you can see it in the slide also. You have this one blue color myosin here, and the thin lines are showing the actins. Okay. So what it says is that when you have an optimal sarcomere length, uh, that time your actin and myosin overlap is perfect, right? And what do you mean by perfect? It means that you have as many number of cross bridges that can form. And if all the myosin and the actins are forming a cross bridge with each other, they all can pull the filament over the M line, right? So the sliding is maximum. There's a maximal force in your contraction. What is this trying to say? Why is this optimal? Because let's say the other two condition, when it is too short means your Z line is very short, right? So it is called over contracted. What is happening? Your actin filament are basically overlapping each other, right? And as a result, because your actin are all close to each other, the myosin cannot interact properly with the actin, and there is very less contraction, cross bridge, and reduced force. So your muscles is very weakened. Similarly, if you're too far apart, again, if you are too overstretched or too long your sarcomere is, then again there will be less overlap between actin and myosin. Again, there will be free cross bridge, and you will have a reduced force. And that is what is being told here. That in this optimal length, which is around 2.2, around 2.2 micrometer, is when you have the optimal length. If you are either too short here, or if you are too long, you have reduced forces, right? You can see you need this actin and myosin cross linking with each other, but it will not happen if they are too close to each other. You can see if it is too small, everything is overlapping each other. So this myosin can interact with an actin, but what about this actin which will not be interacting at all? So you have some actin which are free. They will not getting overlapped. Similarly, if it's too outward, only these actin and myosin can interact. This myosin is free, this actin is free. So again, force is not generated. So you need an optimal sarcomere length. Okay. Okay. Now we come to another very interesting topic. If you remember, we discussed about twitch and strength training and your muscle training, right? So if you remember, twitch is basically was a single brief contraction of muscle fiber. Uh, okay, we haven't discussed about action potential, but I know you already know that all these contractions are in response to an action potential, right? So twitch is basically a brief contraction of the muscle fiber, right? And if you remember, twitch had three phases: latent period, contraction, and then relaxation. Latent is when you have the so the time your stimulus comes and your contraction starts, the lag between that period. Contraction is when your actual contraction is taking place, and then you have relaxation, which is basically them going back to its original length. Now in this, you have two different kind of muscle fibers, as you can see here on your slide as well. Yeah, you have endurance training and you have fast twitch muscle fiber, and we discussed about this very nicely, what all it refers to, right? So let's first talk about slow twitch. Okay, so slow twitch is basically the name given for slow oxidative process. So process in which you are using oxygen optimally and in a reduced manner. Your contractions are slow. You are relying more on usage of, on having many mitochondria, many myoglobin, and many capillaries, right? Because you have a slow oxidative process here, right? But uh, these muscles are they have high fatigue resistance, right? Because they are using aerobic respiration. Like I told, it's slow oxidative process, so it's using the oxidative phosphorylation method for its energy generation, right? Now where do you need this kind of slow training? Where do you need endurance training? Where you have to run faster, right? So you have marathon running. Remember, I told you when you are running for marathon, you have to run longer. So if you need your sustained energy, you need slow twitch fibers, which are type 1 fibers, right? But type 2, there are two types of fibers, type 2A and type 2B. If you remember, type 2A are fast twitch oxidative glycolytic, and type 2B are fast twitch glycolytic. Okay, so type 2A is the in between type 2B and 1. Type 2A has pink coloration, while type 2B are white coloration. So this one is a fast twitch muscle, which is paler type 2A. If it is very, very less red color and more of a white coloration, it's type 2B. Okay. Now these two fibers have very less mitochondria and very less myoglobin, because they want to contract very fast, right? And therefore that's why they have very little fatigue resistance. And what the idea is that you need to do that contraction very quickly. So instead of relying on aerobic process, which will take longer time, in these muscles you have anaerobic glycolysis. You use anaerobic medium, and you also basically use anaerobic conditions for glycolysis and energy generation. Where do you need this? Where you need to act very fast. Where? Short or powerful movement. Where do you have this? You have this in weightlifting, you have this in sprinting, which are very quick races. So for there, where you need more energy, those you train them for strength. They are fast twitch fiber. Where you train them for endurance, they are slow twitch fiber. So these are the basic differences, and we have done this. We have discussed this in the problem solving session also. So you can go back to the session and look into this in more detail. Okay, the next topic. So like I told you, I have already told you that all these are in response to action potential. So we will now look into the action potential part, where it is basically talking about signal transduction. And we already know about signal transduction. They are there because of the excitable cells. Cells which can get excited are basically cells that can relay action potential. And those action potential are basically signals which can be relayed to the next in line so that the signal is carried to the respective cells or tissues or muscles in that case. Right. So let's discuss about this in a bit more detail. So let's first talk about action potential, and then I will also talk about its Hodgkin-Huxley model. And action potential and Hodgkin-Huxley model are basically talking about action relaying, basically action potential relaying and propagation. So to formally define action potential, it's basically change in your electric potential across neuron, electrical potential because of the ions involved. So this can be across neurons, or as you will see later, it can be also across muscle cells, right? Now, as you can see here in this slide, you have something called stimulus. You have something called field initiation. You have depolarization, repolarization, hyperpolarization, and resting phase. Okay. So let's begin with this resting phase, which is basically resting potential. Potential is basically difference in the ionic concentration, right? So at resting stage, when your cells are not excited, they have a potential around the so at the membrane level they have a potential of minus 70 MB depending on the sodium and potassium ions concentration, right? uh, we also remember we have channels for all these ions. You have sodium ions, you have potassium ion channels, and or they are also called pumps. So these pumps operate to maintain the flow of sodium and potassium. They let the ions in and out of the cells, which basically determines the concentration of the cell around the membrane and the ionic potential, right? So at resting stage, as you can see here, the action potential, the voltage or your concentration is around minus 70 millivolts. Okay. The next phase is, first thing is depolarization, right? Now, what is depolarization? It basically means moving away from this resting stage, right? How does that happen? Your sodium gate open up and your sodium channels opening leads to sodium ions moving inside, right? Sodium ions are positively charged. As soon as they enter inside your cell, inside your muscle, your positive ion concentration increases. So whatever was your minus 70 will now start going positive. Now, uh, you have certain ions entering. Your voltage or your ionic concentration will, of course, reduce, become more positive, but you need a certain threshold that has to be crossed for it to generate an action potential. And you need to cross minus 55 volt going towards a plus 40 millivolt for the cells to become depolarization. Means that many sodium ions has to enter the cell. Only then will it be called depolarized. Then your repolarized means to coming back to your normal condition, or to basically the opposite trajectory to come back to the normal phase. So at that phase, at repolarization, your sodium channels will close. They will get inactivated. But whatever sodium has come in, they will be now compensated by potassium ion. So this potassium ion, sodiums have moved in. So potassium will move out because sodium and potassium inside were contributing to the positive ion charge. So if you push out another positive ion from the inside the cell to outside, again your membrane will start becoming negative, right? So your potassium will move out and the cells will again, or the membranes will again start restoring its negativity. So that's why from plus 40 it will start getting down. But what happens is it becomes hyperpolarized. So why? Because your potassium ions will, so sodium ion open, channels open, sodium enters, channel closes, and then it stays there. But potassium ion channel opens, but for closing it takes time. So during the lag of closing, you have more ions which have left out, which have exited, because of which instead of coming to minus 70 it goes down to minus 90, minus 100. So it becomes hyperpolarized, right? So your membrane becomes way more negative than it should, and then the next phase is basically from that hyperpolarized state it has to still come back to the minus 70, right? So at this stage what happens is your pump comes into action, and these pumps do the opposite of what happened during your rest in your depolarization. So at pump, when my hyperpolarized states, your pumps get activated. I'll tell you why are they pumped. But what they do is they push sodium out and they bring potassium in. Okay. So when they bring potassium in, again your membrane starts becoming towards higher end from minus 90 to minus 70. And these are pumps because they use ATP. They need to use energy to open up and do this whole transport in and out, right? So it's beginning with more sodium moving in and potassium going out, but because more potassium goes out, your pump has to get activated to bring it to this minus 70 millivolt, and that's what your action potential is referring to, this image here. And the next thing, which was Hodgkin-Huxley model, is basically a model which is explaining how these potentials are generated and propagated. Remember, they gave us the mathematical formulation of these propagation, but they had no idea about the channels, right? So you will not see that they have incorporated the information of channels, but they have electrically or electrophysiologically determined how these action potentials are generated and propagated, right? And if you remember, they did this from a giant squid axon, right? So basically they are talking about the whole action potential movement as an electrical circuit, where, if you remember electric circuit, you have a capacitor, you have a resistor, and you have a battery. So your capacitor is the storage of charge across the membrane. Your resistors or your conductance is basically the sodium potassium channel, sodium potassium ions, and your batteries are basically equilibrium potentials for each ion. So whichever condition this comes weight to. So basically, it's just saying that how these cells in a circuit are behaving, or how these circuits are behaving in with respect to the biological circuit, right? And you have this formula which was given by them, which basically is giving an whatever I just spoke to you about hyperpolarization, depolarization, repolarization, that is given in a mathematical term, and it basically tells you that biology can also biology, physics, chemistry, everything is interlinked, and you can explain all the methods that you see biologically with very good physical laws. Okay. Right. So you remember the Hodgkin-Huxley models as well. Next, like I told you, so this action potential is generated, let's say, in neurons, and these neurons are, as you remember, they have to conduct this information, relay the information across this entire body. So that the relaying is called propagation. And in the neurons, you have either saltatory movement, if they are myelinated. If they're non-myelinated, then they take it slowly. So you can have myelin sheath, which basically has this jumping conduction. If not, they move slowly. And we have seen we need both slow movements and faster movements. But when they reach the end of the axon, they have basically chemical synapses that take place for your relay of information across neurons, right? So you basically have these, let me tell you. So your action potential reaches the hillock or the end. And here what happens so this is your, I told you, right? This is your action potential. So this is the neuron which has the action potential. This is the next neuron that has to start the action potential. And the gap between them is called synapse. And this has to be covered. This is how it has to go to the next one, right? So at the terminal, when the action potential will arrive, what happens is you have voltage gated calcium channel. What is voltage gated? You basically means that depending on your signal, that gate will open up. So your calcium channel opens up. As your calcium channel opens up, you have calcium influx, right? Now, when your calcium ion is into your cell, it will trigger something called neurotransmitter release, and the neurotransmitter vesicles will fuse with the membrane, open up, and it will be pushed into the synaptic cleft. At the synaptic cleft, when you have all these neurotransmitters will bind to the receptor at the postsynaptic neuron. And as soon as that neuron binds to the neurotransmitter and it knows that, okay, I have to propagate this signal again. So I now have to generate signal potential. So what do I do? As soon as I bind to neurotransmitter, my sodium ions channel will open up and my sodium channel will rush in. And then the entire process of action potential takes place there. right? So you have this chemical influence here. So called as calcium and a neurotransmitter, they relay the information to the next neuron. So you have to remember the synapses, you have to remember the neurotransmitters, and you have to remember the importance of calcium ions here. Okay. Okay. So after we did this, we did about, I just told you about transmitters, right? Now, the transmitters which are released by the neurons can be of two types: excitatory neurotransmitters and inhibitory neurotransmitter. The names themselves tell what they are trying to do. And if you broadly remember, we discussed about excitatory neurotransmitters, examples as glutamate and acetylcholine, and inhibitory as GABA and glycine. Okay, so these are two main types of neurotransmitters. Um, Along with them, you also have epinephrine and norepinephrine in excitatory, and serotonin and dopamine in inhibitory. Now, what do they do? So these excitatory ones, they are basically the ones which make the postsynaptic neuron to generate action potential. So you can remember it like this. These are excitatory, so they're exciting or they're causing excitation in the next neuron they are interacting with. While the inhibitory ones, these prevent the postsynaptic neurons from generating an action potential. Right? Now, what do they do? So excitatory neurotransmitter, they increase the transmembrane ion flow of postsynaptic neuron. This is what I told you. But inhibitory ones, when they bind to the next neuron, they will decrease this transmembrane ion flow, and your this movement of ion stops. And because it stopped down, the depolarization becomes difficult. And along with this, they will also open up potassium channels, so making it even more difficult to. What will happen if potassium ion channels open up? Potassium will move out and it will become more negative. So that's why it will not generate action potential. It will be difficult to reach the threshold level, right? So yeah, so inhibitories are basically that stops the action potential generation. Now you should understand why. I told you something about hemostasis, which is basically keeping your body in check, keeping all the body organs functioning similar in a normal way, balanced way. Now, if you have excitatory neurotransmitter, which is everything is conducting and relaying action potential, there needs to be someone that keeps a check on this, and that checking is done by the inhibitory neurotransmitter, because that will delay the action potential generation. So you need the inhibitory neurotransmitter to reverse the activity of the uh, positive or excitatory neurotransmitters. Okay. Now, after we did this, this is where I think we had finished about your neural system, muscular system, and we will do, I think, briefly once. Yeah, we will just touch upon neuromuscular junction, but towards later. Now we are going to talk about your associated problems with not functioning of your nervous system in a proper way. So what all problems can you see if your nerves or neural system doesn't function appropriately? But to understand that, we had a brief we just took a brief look into the brain anatomy. And if you see here, your brain has divided into several lobes, and has different parts, and all these different lobes or different parts of your brain are responsible for different kind of function. For example, cerebellum is mainly for your motor function, so your voluntary movements, posture, balance, while your cerebrum is mostly for your higher mental function. It can also be part of different kind of motor function. They have Broca's area, which is in temporal lobe. They are associated with sensory function. So all the kind of activities that we do, parts of your brain are responsible for mitigating those functions, right? Okay. Now, I told you we have discussed about several disorders, several problems associated with brain. So let me talk about them quickly without spending too much time, because we have done that already in the respective session, right? So the first thing is epilepsy. So epilepsy, as we know about in simple terminology, these are seizures. So basically, epilepsy is when you have, it's a neurological disorder when you have recurrent, uncontrolled seizures. And this is because of abnormal electrical activity in the brain, right? So in your brain, there is this, as you can see, it's a hyper excitable state of neurons and the neuronal network, because of which you have too high electrical activity that leads to your uncontrollable shaking in your body, which is called seizures, right? And this is because there is an imbalance between your excitatory and inhibitory neurotransmission. So like I told you, your inhibitory neurotransmission should be following your excitatory, but there's a lag here, and the two neurotransmitters which are responsible for epilepsy are glutamate and GABA. And what happens is that because there's an imbalance between the concentration of these two neurotransmitters, your neurons are hyper excited, and you have this extreme shaking in your body. So you have these convulsions, and because your body gets too tired, and your cells are hyperactive, you end up losing your consciousness. And you also have several associated problems, like you have sensory disturbances, you seem to be in a fuzzy state, right? So yeah, so that's what is epilepsy. So you can remember epilepsy as seizures, and which is basically hyperexcitable state of your neurons, right? The next thing that we discussed was memory. And if you remember, we discussed that we basically, what we meant here was, how do you have the memory generation? What is memory generation, first of all? So memory is a process by which information that you are seeing around you, or hearing around you, or listening around you, they are encoded, they are stored in your brain, and at at the required time they can also be retrieved in your brain from, right? So you have sensory memory, like I told you, you touch something, you see something, and you store it. That's milliseconds. It's a very short memory, uh, or rather very short memory. Then you have short-term memory, which are basically seconds to minutes, which work in seconds to minutes. And then you have long-term memory, which are, like takes days and years, are stored in days to year basis. And all of these types of your memory are stored in different places. For example, short memories are in prefrontal cortex, long-term memory are in hippocampus, and then it goes to cortex, right? So you need to remember about hippocampus, amygdala, prefrontal cortex. I'm not talking about detail because we have other topics also, but you need to remember it like this. And the idea is, the stronger your synaptic connections, the more stronger your uh, memory is, right? So all these cortexes that I just spoke about, and this is how I was telling you, you have stimulus, sensory memory, quick memory, short memory, and then long-term memory. And then you need to retrieve that also. So this encoding process is dependent on what is the length, or what is the strength of the synapses between your neurons. Okay, right? And then connecting to this, we have two terms: long-term potentiation and long-term depression. Quickly to explain you, long-term potentiation is basically a long-lasting increase in your synaptic strength, and which is following your high frequency stimulation of a synapse. So you keep your synapse is basically stimulated very frequently, and it builds up the or it increases the synaptic strength, right? And this mainly happens in your CA1 region. So as you can see here, you have these different this is a terminal this is a pre-synaptic. This is one axon, this is one, or this is one neuron, and this is one neuron. And the more they interact with each other, the more strong their relationship becomes. So basically what happens here is, if I just have named these receptors, you have very high frequency of stimulation. They are stimulated again and again and again, and your, as in general action potential, the most frequency of stimulation, more calcium influx via these NMDA receptors, and this will activate these kinases, and this will result in the insertion of AMPA receptors, and that will make a postsynaptic response very strong, right? And this, because as you can imagine, the moment I was telling that you have to frequently stimulated, it tells you how do you learn stuff, because you are repeatedly telling a particular thing and repeatedly um, like actively thinking and listening, so you're learning and putting it into your memory. So that's long term potentiation. And the other side of this will be long term depression, which means that this is a long lasting, this is over a long period of time, you have this synaptic string which is becoming weaker in time. That's because these neurons are not at all or very low, at a low frequency they are stimulated. So because there is low calcium channels, calcium channel and activation, low calcium entry, instead of kinase they activate phosphatase, and these AMPA receptors which are responsible for memory are removed. And what happens here is this is for your forgetting. So you start forgetting stuff. You have your synaptic balance is disrupted. So all of these are part of your long term depression, right? So you have low frequency stimulation, and this is when your cell becomes less responsive, so glutamate, right? Okay. The next one that we discussed was Alzheimer's, and the book here, like I told you, talks a lot about how Alzheimer's actually takes place, and this is how you can see the difference between just by seeing the image itself tells you how your Alzheimer patient's brain looks so different than your healthy person, right? You can see that an Alzheimer's patient. I know people are aware what otherwise Alzheimer's is called. It's called dementia. You have memory loss, and not just memory loss, you have communication problem, because of course you can't communicate properly, you are not understanding stuff, you have difficulty, you have confusion, you have very much problem with what is in and around you, so your vision and spatial awareness. So things which was very simple for you and you knew them every day would become unknown to you, because you have problem with retrieving the information, right? So the Alzheimer's is basically, you so how is it different from your epilepsy? Epilepsy doesn't talk about your forgetting or talks about memory. Epilepsy is your electrical activity of brain. Alzheimer's talks about your memory. It talks about your memory loss. It talks about cognitive decline. It talks about behavioral changes, right? And that's because of so as you can see, it is because of these deposition. Remember, we spoke about plaques, we talked about tau protein, we talk about neurons damages in hippocampus. So all these together leads to memory loss, right? So when your part of your brain starts getting plaque deposition, you have this protein aggregate forming. So it disrupts the neurons there, and because neurons are disrupted, your memory loss takes place, right? So Alzheimer's is a memory loss associated problem. The next one that we discussed was Parkinson's disease, where you have these patients are characterized by excessive shaking of their hands. So you have tremors. You have the main problem here is the diminished substantia nigra, and you have these neurons which are problematic. Along with the shaking, what you see, they have ptosis, so their eyelids it's closed in one part. They have constipation, they have high sweating, they have depression, they have sleep disorders, right? So a Parkinson's disease is basically a motor dysfunction. Okay, it's a dysfunction. That means the movement that was under our control is no more under the control of yourself. So it's a motor function problem, right? So Parkinson's is a motor function problem, and this is because of your dopaminergic neurons. Now, what we didn't discuss is what is a neuron which is called dopaminergic or cholinergic. The basically you name a neuron based on the type of neurotransmitter it releases. So if it releases dopamine, it is a dopaminergic neurons. So in Parkinson's, these dopaminergic neurons are dying or are disrupted. There is loss of dopaminergic neurons, and as a result, whatever downstream dopaminergic neurons were there, they are also getting affected. You have dopamine inefficiency of your body, and that's why you have all these different associated problems. Now, because it's a dopamine problem, you don't have this neurotransmitter. These problem can be treated by L-DOPA or synthetic dopamine precursor, right? So this we discussed that we have drugs that can be used to treat this kind of disorder, but at the root of it, at the heart of it, this is a motor dysfunction uh, related disorder. Okay, right. So we discussed all about neural system. We finished discussing about the problems associated with neural system. Now we will talk about what we had discussed in week 8, which was about stretch reflex arc. Okay. Now what is stretch reflex arc? It very beautifully connects whatever we have been discussing so far. And what have been discussing we have been discussing so far? We're discussing, so we have discussed about neural system, we have discussed about action potential, and we have discussed about muscles, right, contractions. This stretch reflex arc puts it all together. Now on your image, on your slide, you have this person who is touching candle, and it takes no effort to guess what this person will be doing in another split of second. He will be removing his hand, right? So similarly, there's something called knee jerk reaction. If you know, if you tap your knee with a hammer, without any effort, your knee will move forward, right? Or if you touch a pointed object, let's say there is some needle and you touch it, you immediately remove it. So you have a reflex action and you have a stretch reflex. This is a response which is which is taking place due to the perception that you have had. Now, what does that mean? That means, in simple terms, first of all, that this person here is touching the source of it, which is candle. So he is basically touching the candle, feeling the heat. This is excessive heat. So your finger receives this information. Your afferent neurons, which are the sensory neurons, these take information to the brain and spinal cord. And there they process the information and says, hey, you need to remove your hand from or finger from that part. So it will now give you information to afferent neuron, which is a motor neuron, and this information will be relayed to the muscles. The muscles will then contract and he will remove his body, remove his arm. So this contraction is because of this entire process that has taken place. So in other terms, you can see it like this. You have a stretched muscle which is doing some activity. The stretched muscle takes it via one neurons to the central processing unit. And from there, you have the response coming, which goes back via motor efferent neurons to the stretch muscles to contract it. As simple as that. Stretch, reflex, arc, right? And these are the different neurons that are part of the circuitry. So you have sensory neurons and you have motor neurons, as you can see the name here. Gamma motor neuron, alpha motor neurons, sensory neuron. Do you know what are these IF and EF? These are intrafusal muscle fibers. These are extrafusal muscle fibers. And we discussed this, I think, in 2 sessions. These muscle fibers are basically present with a respective set of neurons. Sensory neurons detects a stretch, takes the information to the processing unit, which is brain / spinal cord, and the motor neurons bring the information back to these muscles, which are extrafusal muscles, and these are the muscles that will send the information to the or that will do the motor movements, right? So that's why they are motor neurons, and that's why they are sensory neurons. And what you need to remember is which one is present where and how are they moving. And also you need to remember this afferent and efferent name, and I had given you a cue of how to remember this, so you can go back to the problem solving session and see how they are related, right? Okay, now your all these things is taking place, right? You have these stretching taking place, you have this contraction taking place. What exactly is taking place then? What is happening? You have, this is where we are talking about neuromuscular junction. Now, as the name itself suggests, that we are going to be talking about neuro, so whatever we have so far read about nerve, and we will be discussing about muscular more response. So your nerve or neural circuitry and muscular contraction, how are they connected, right? Now, as you can see, this is your axons, or this is your neurons. Now, after a long line of neurons, it will finally come to the muscles, right? Now, the information has to go to the muscle. The muscle has to respond now that, okay, let's say the neurons are dopaminergic neurons, and it says, hey, you need to contract. So, or you need to contract now to move your hand away from the fire. So that information is now being relayed by this action potential to the muscle. And you see there are so many players here. First of all, you have the uh, sorry, you have the acetylcholine vesicles, you have the neurotransmitter vesicles, you have the voltage gated sodium channel, you have the uh, on the muscles, you have the motor and muscle fibers, axons. So we will now be telling what exactly is it saying, and the procedure or the process which covers this part is called excitation, contraction, coupling. If you remember, excitation is basically your ions in your neurons, which is excitation. Contraction is the response of muscles. So excitation, contraction, coupling, which basically mean they are taking place together. So what is happening here? So you have this electric signal as action potential coming to the muscle fiber and leading to the contraction. Now, if I have to tell you step by step, let's just begin from the first step, which is basically a motor neuron. So anything that takes information to muscle is called a motor neuron, obviously. Now, this motor neurons uh, has the action potential traveling to down it, and it takes it to this synaptic junction. Okay. Now, at the synaptic junction, you have the action potential releasing calcium ions because of the signal. And as your calcium ion is released, you have the vesicles which are with the neurotransmitter fusing with the membrane and breaking open. And here it releases something called acetylcholine. Acetylcholine is released into the cleft. Now, once your acetylcholine is released into the cleft, it will bind to its receptor. What is the receptor of acetylcholine called? It's called nicotinic receptors. So these nicotinic receptors, which are present on the motor end plate of the muscle, they bind to the acetylcholine. And what should it do? I told you it should also generate action potential. So it will open up the sodium channels, and that will lead to the depolarization of the muscle cell, and the muscles will now have action potential generated. So it's basically activation of the muscle fibers. Now, once your muscles have an action potential in them, that action potential will spread through the muscle fiber, through the transverse tubules, and then again it will reach to the calcium, so the sarcoplasmic reticulum, and it will reach to calcium ions release. Again, when your calcium ions are released, what happens is you have these voltage sensors which are called the DHPR, remember, DHPR receptors. These get activated, followed by ryanodine receptors, and they again release calcium. Okay, now you have these action potential, ultimately again leading to calcium release. Now, what is calcium doing? Calcium binds to troponin C. I told you, where did we discuss about troponin? We discussed about troponin and tropomyosin, actin myosin sites. So when your calcium is released, it leads to the binding of, or it binds to troponin C, and then it leads to myosin and actin binding, and the cross bridge is forming, and your muscle shortens up, right? So your contraction is taking place. And as soon as your action potential finishes, that means as soon as the impulse for generation of action potential is stopped at the muscle, at the motor end plate receptor site, your calcium is pumped back into the sarcoplasmic reticulum, and everything goes back to its original state. So you see how from the release of acetylcholine, which is due to an action potential, you lead ultimately to your contraction that we had studied by sliding filament theory. They are all connected, is what this whole process of excitation, contraction, coupling trying to say. Okay, right. And this was where we ended this long topic of neural system and skeletal system and muscles, and we connected all the dots together, and we saw how they are functioning as a unit, right? Hence, moving forward, we will be now looking at the other systems which are part of the organism. right? Right. So the first system that we had discussed was hearing system. Now, as you can see, we have discussed about hearing systems already, right? We know that they have the middle ear, the outer ear, and the inner ear. You have outer ear, which collects the airborne sounds. Then you have the middle ear, which does the conductive mechanism, which conduces or which conducts the information. And then you have the inner ear, which basically converts it into nerve impulses, right? So as you can see here, all these three things that I just read are basically mapped out here. So you start with this. So your sound waves appear at the tympanic membrane, which is here. Okay. These are the inner ear malleus income steps. Okay. So you have the first thing is sound which reaches your tympanic membrane. Uh, what it does is the tympanic membrane will move. As it moves, it causes displacement in these auditory ossicles. Ossicles means bones. So these bones will move. Now, as your bones or as your malleus stapes move, so the last one, which is stapes, as it moves at the oval window, it establishes pressure waves in the perilymph of the vestibular duct. So this is a duct. As it moves, it starts a waves here. Okay, these are move. They have generated waves. So they are now moving further. Now the pressure waves which are here, they distort this basilar membrane on their way to the round window of, so they are moving like this, but on their way they are, how do you say, they are basically disturbing the basilar membrane, the membrane which is lining at the covering, and that vibration of that membrane causes the hair cells vibrating again the tectorial membrane. Now at the end, what is happening is information about this region and the intensity of the stimulation is relayed to your central nervous system over the cochlear branch via the nerve which is here. So you need a neuron which takes it to the brain, right? So this movement here leads to the activation of nerve system, and that's how nervous system takes the information to the brain. So all ultimately what happens is a sound wave which is coming here propagates into an information in the inner ear, and that takes the information to your brain for processing, right? So here also you have your nerves, which is playing a crucial role. Okay, right. Next, we read about olfaction system. Now, olfaction system was basically where we were talking about smell. So it is basically your sense of smell. And remember, we spoke about different odors which can be detected and processed based on what is the intensity and where and how they are perceived, right? And basically you have these odor molecules, and they will bind to the receptors on your neurons, and as they do that, they will activate a cascade of signaling which will again generate action potential for you, whatever movement you have to take place, right? And that's why if you see this figure here, you have all these olfactory bulb, epithelium, these odor molecules, and then inside you have the axons, the odorants. This is the odorants, the receptors. Ultimately they have the axons, so the neurons get activated and pass this information ahead, right? And so limbic system, what does limbic system mean? Why is limbic system connected here? So because your olfaction system is connected to the limbic system, you have a very strong link between smell, emotion, and memory. What does it mean? So let's say I'm smelling something good, and I like that smell. So I associate it with something. Let's say it's coming out of a pie, apple pie, and I like that smell. So that smell, I am perceiving that smell. First of all, I am behaving in a good manner. I am like enjoying it. And I will save this in my memory such that next time I smell this, I know that this is a good food and I should run towards it. Contrast, when you are near a garbage, you smell it and you move away. That's also stored in your memory. So next time when I smell that, or whenever I am standing near a garbage, automatically the smell initiates a response of me moving away. So that's what is your limbic system doing. Your emotions and your memory is part of limbic system, and this presence of limbic system so close to nasal cavity is what makes your connection between smell, emotion, and memory so strong. Right? So basically, you have these all these olfactory epithelium bulb tract, which are all different parts of your olfactory system involved in detecting your odorants or odor molecules and taking it to the brain, and then perceiving the information and then acting upon it accordingly. Okay. Now, how many odors can you smell and how many times you can perceive it? We've seen this all in the problem solving session. So I suggest you go to that session to look into this in more detail. Okay. Right. And next week, which is week 9, we move to the visual system, which is about eye, which is about process of seeing. How do you see? How do you perceive an image? How do you first of all form an image? All of these things, right? So if you see here, first of all, eye system, any system that we have done, we have started by looking into the anatomy of that system, right? So in eye system 2, we saw the retina, we saw the iris here, we have the sclera, which is in front, you have the cornea, which is here, then you have the pupil, lens, which is covering ciliary body, conjunctiva is a retina where the image is actually formed, vesicles, which are taking the blood vessels, which are taking the information to the brain. So all of these we have looked into. But what is most important is about the formation of image in eyes, right? So how is our image forming in eye? And you remember we spoke about how the eye is similarly functioning similarly to a camera, right? So like in camera, you have your light rays which are focused onto one part. Here in the eyes, it is the retina, which is to form an image, right? So all the other structure that I spoke to you about, where you have the cornea, you have the pupil, these are all responsible playing and role in the image formation. So you have the cornea, right? So cornea is where you have your light entering from, right? So you can see here you have your cornea and your light is entering here, right? So the first thing is light is entering through cornea and they will bend the light. So most of the light gets reflected, right? Then how much of your light enters is regulated by your iris, which controls your pupil size. So pupil is basically controlling how much of your light should enter eye, and you have the iris, which controls the size of the pupil. Then you have focusing. So your lens, they change the shape to focus the light precisely on your retina, right? So you have, if you have distant objects, far objects, your lens becomes flat, your ciliary muscles become relaxed. If you have a nearer object, your ciliary muscles contract, lens becomes rounded. So you basically what you want is your light rays to focus on the retina, right? And then on your retina, the image formed is real, inverted, and a smaller one, right? And along with this, your retina has these photoreceptor cells, rods and cones, which basically convert this light into electrical signals for you to perceive the image, right? So your image is formed here, but all these optic nerve and all these information on the image formed is coded by the rods and the cones, and that's for light vision and darker vision, and they are taking the image to the brain, and your image is being perceived there, right? So coming to electrophysiology and photoreception, basically, so in your when you talk about electrophysiology, you're basically talking about the electrical activity of the retina and how it is taking place. And you have this, as you can see, remember we told about resting potential here, and this is not the usual conditions here. So in your dark phase only, you have this threshold reaching when it's dark only, and then suddenly you have light coming and you start having these waves formation, right? So you need to remember this difference between the electrophysiology and the photoreception with respect to a visual system. I'm not going into detail considering the lack of time, but yeah, please do look into electrophysiology. It is not following the usual pattern that we have been studying so far. And coming to photoreception, where we are talking about rods and cones, we are basically talking about rods which are used for night vision, and the pigment in rods and rhodopsin, and cones which are used for daylight vision, where you have the photoopsins. So all these rods and cones, darkness, light, together they're responsible for something called phototransduction, which is basically what is electrophysiology. And these rhodopsins that take light, they change. So sodium channels and like the movement of sodium ions are here also, but what you need to remember is how do they start. So just keep a check on how are these formed. Okay, how these uh, ions, how these election potentials are generated. Okay, right. This we are short on time. I will move to the next topic, which we have discussed only very recently in the past last week, right? So we are talking about cardiovascular system here, which is basically your heart and your pumping of your blood by heart, right? So first of all, you need to remember the anatomy of heart, which we have done multiple times. What I told you, if you remember, was whatever you see in your figure, the right side on your slide, if basically what I mean is this part of your heart. So you need to imagine this is not this side of your heart. This is basically, so if you imagine this part is this side and this part is this side. Okay. So why is this important? Because you need to remember which side of the heart is responsible for what kind of activity. And what do I mean by what kind of activity? Which type of blood flows through that part of the heart? Is it the deoxygenated blood or is it the oxygenated blood? Okay. So as you can see, the right side has the deoxygenated blood and the left side has the oxygenated blood, right? The flow of the blood is all across the body. What is above lungs, above heart, you have the lungs. What is below heart, you have the other parts of the body. So the circulation of blood below heart or the other parts of the body is called systemic circulation. Circulation of blood to and from lungs is called pulmonary circulation. Anything which is pulmonary is heart, it's lungs associated. Anything which is systemic is body parts associated. Okay, so you have pulmonary circulation and you have the systemic circulation. Now, very simply, what is the purpose of heart? Heart is just collecting blood and putting out blood. Okay, so our heart will have vessels which collect blood and then vessels which pump out blood. First thing. Second thing, it has four walls, four regions or four halves, and two parts, left and right. Each part has upper part of the heart is called atrium, lower part or lower sac is called the ventricle, right? So these this is your ventricle, this is your atrium. This is, like I said, this is your right, this is your left. So this is your right atrium, this is your left atrium, this is your left atrium, this is your, right atrium, right ventricle, left atrium, left ventricle. Okay. Now in these four sections, the part of the role of atrium is just to collect the blood and pass it to ventricle. And the role of ventricle is to pass the blood out of heart, right? Where the blood goes to and where the blood comes from, that is what determines your side of your heart. Okay, now let me tell you how is this happening. So let's first start with this, which is your vena cava. Okay, so vena cava is the main vein. So you have blood vessels of two types, which you will see in details in a while. But for now, remember that you have veins and your arteries. The biggest vein is vena cava. The biggest artery is aorta. Okay. Now veins, the biggest vein is the vena cava. So the first thing that happens is via the vena cava, you have the deoxygenated blood going into the the right atrium. Now, from the right atrium, it goes to the right ventricle. Now, from the right ventricle, it goes to lungs. Why? Because this is a deoxygenated blood. Means what? It is rich in carbon dioxide. It has low oxygen. So it needs to be purified. It needs to push out the carbon dioxide. When exhaling, you are taking in oxygen, giving out carbon dioxide. Those carbon dioxide is coming from here. In this blood, you are more of carbon dioxide. So this blue blood, this is blue because of that reason, that you have more of carbon dioxide. So it needs to go to your lungs and the lungs will push it out. Right. So since you have vena cava, which is pushing your blood into your atrium, and you have this uh, blood vessel which is taking blood out of heart to the lungs. This is a artery. Now what does it mean? Veins in general will take blood into the heart, and arteries take it away from the heart. I told you, remember it as a artery, a vein. So artery takes blood away from the heart. Now what is happening here is, it is taking blood away from the heart, obviously, but it is taking to lungs. So that's why it's called pulmonary artery. So blue color has only, mostly it is associated with veins, except for pulmonary artery, which takes away the blood, the deoxygenated blood coming out of heart to the lungs. Now the opposite, now whereas the vena cava getting the blood from? Vena cava is getting the blood from the entire body cells. All the body tissue which is respiring and giving out carbon dioxide, the veins are collecting those blood and bringing it to the heart. Now the opposite. Now you have oxygen rich blood, which is the oxygenated blood. So where will you have blood which will be rich in oxygen? That will be coming from the lungs, right? So this is what is happening, right? So you have your blood which enters your left atrium, as you can see here, right? So your left atrium, left ventricle, and pushing out to body, because you want oxygenated blood to the entire body. So anything that takes blood out of heart is called your artery. So the one that takes it to all the blood body cells is the aorta. So aorta is the largest artery. That makes sense. But then with what brings it? What brings anything that brings blood into heart, like I told you, is a vein. So the blood vessel that brings blood, oxygenated blood from the lung, because lungs is where you have carbon dioxide outside and oxygen coming inside. So your your blood is now rich in oxygen, that is being carried by a vein into the heart. So anything which enters the heart is a vein, but because it is bringing from the lungs, it is called pulmonary vein. So veins in general have the oxygenated blood, except for pulmonary vein. Arteries in general have oxygenated blood, except for pulmonary artery. And that's how, if you make this connection, uh, the exceptions become clearer to you. Okay, so that makes life easier for you. Now, along with this, um, every time you have these, so this is the entire cycle, right? You need your blood to enter and then move into ventricle, move out of ventricle. So all of these has to be ensured in a unidirectional manner, because everything is governed by pressure. Okay, everything is governed by blood filling up the chamber walls are like inflating up, putting pressure on the walls, and then you have to form them. Because of the pressure, the channels open up for the next chamber and it moves out. It cannot come back. So at every such ejection point or every such opening, you have checkpoints, and those checkpoints are valves. Okay, at every point you have valves which open up and then close back, so that blood is always moving in a unidirectional flow. And the movement of one blood from one part to the other part is what you measure as your blood pressure, and that's called the systolic diastolic pressure. So when you have, so and everywhere, whenever you have a muscle and whenever you have a contraction of muscle, you have these movements taking place. In heart also you have muscle, cardiac muscle. These muscles contract, and this is what all leads to the flow of blood. So when you see a cardiac cycle, which is basically all this pumping of blood taking place, you start it with atrial systole, right? Which is what atrial means, the upper chambers, and atrial contraction is atrial systole. So the first thing is atrial systole, so which is basically atrial contraction, which is forcing a small amount of blood into the relaxed ventricles, right? Now, as your atrial systole ends, which is basically all the blood is pumped into the ventricles, your atrial diastole begins. Now, what is diastole? Diastole is when you have relaxing of your atrial muscles, right? Now, because ventricles are filled with blood, these start contracting. So these are the next phase is ventricular systole. So you have ventricular contraction now, and you have the atrioventricular valves being closed. So the valves are generally named on, for example, AV valves are present in atrioventricular junction. Okay. So the first thing is ventricular contraction pushes AV valves closed, but does not create enough pressure to open semilunar valves. What is semilunar valves? These are the valves from the blood vessels where the ventricles will push the blood into, right? After a sustained period of systole, the ventricles will then, after a sustained period, the ventricular systole will actually take place and they will force the blood open, push them out into the blood vessels, and because of the opening of semilunar valves. Again, your diastole will take place, which is basically relaxing of your ventricle muscles, and the blood will flow into the semilunar valves, and the valves will close down, and then will move it to the blood vessels wherever they need to go. And then you have the ventricular diastole finishing up. So all the muscles are now relaxed in the heart. Again, you have blood going into the atrium, everything starts. So all this cycle, as you see, is all taking place in 800 milliseconds, not even a second. So it's very quick. Yeah. And this entire thing is called cardiac cycle. And whenever you have a stethoscope on your doctor's ears and they're checking in your heart, what they're looking for is lub and dub sound, which is basically the closer of these valves. So all this bicuspid, tricuspid valves that we know of, atrioventricular nerve. So these closer of your valves basically tells you that the blood flow in your heart is taking place. And that is what is generating your pulse as well, that we also check for your that also your doctor checks for your health. So all these functioning is what is being checked upon as a measure of your health, right? Now, again, because we have been doing it so far, this system also has its own electrical activity, and that is governed by cardiac conduction system. And we have discussed it that this movement of blood or the contraction of muscle is because there has to be something which generates the impulses. So in heart, you have the sinoatrial node, which generates this space. And there is a series of impulse generation and propagation that takes place to ensure that the blood is effectively pumped by the heart. So you have the sinoatrial node which generates the impulse then reaches the atrioventricular node, which is here. From there, it connects. So the atrioventricular nodes is basically connecting your atria and ventricles. So from there, you have these bundle branches through which is present between the ventricles. So from here to here, from here to these different branches. And lastly, they have the Purkinje fibers, which are basically for the ventricles. So they will depolarize here. And we read about lag here. So there is a lag in the conduction. It's a slower conduction to ensure that the entire blood is pumped out effectively, right? So be careful when you are reading about conduction system. You just need to remember which node is present where, which node starts the impulse, and what is the delay here. Again, a slight like unique feature of this system, so you need to be clearly remembering that. And lastly, this is the kind of image that you would have seen if people do check the heart activity. It's called electrocardiogram, which basically is reflecting all these different stages that we just read about. For example, we read about, so as you can see, so this is the kind of graph that you see. You will have a wave, P wave, QRS, and T wave. So your P wave, which basically is reflecting upon, which is basically showing the electrical impulse when it has moved to the lower chambers of heart. So you start with ventricular systole, right? And then you have this interval, P and R interval. This basically talks about the ability of the wires to conduct electricity from upper to lower, because what we mean here is that this is a lower chamber contractibility. What is the length of this contractibility, how quickly this signal has come to the ventricles. Then you have a QRS, which is basically conduction of electricity through the lower chambers. Why it just shoots suddenly? Because you remember, this is a at relaxed state, and then suddenly it starts the pumping because it fills up the blood. That's why it is up and down, and then again it goes back to repolarization. So you start with ventricular depolarization, then you have the the lower chambers excitation, and then again you have the repolarization. Okay. Yeah. So in the shortage of time, I am not able to explain in detail, but yes, we discussed about electrocardiogram, so you can see in the problem solving session. Okay, the second last week of the series discussed about respiratory system, where we basically looked into the uh, exchange of gases in the bloods, right? So this is how your respiratory system looks. We discussed about the upper respiratory tract and the lower respiratory tract. We have the nose, the pharynx, the larynx, and then you have the entire lung with their bronchi and trachea, right? Now, when we looked into the respiratory tract, we saw that our respiratory system, we saw that how the blood has different concentration of gases, and those are responsible for your exchange taking place, right? So the exchange of gases is governed by partial pressures. So partial pressure of oxygen and partial pressure of CO2. Wherever the partial pressure is higher, let's say carbon dioxide is higher in, let's say, a blood vessel, then carbon dioxide pressure is lower in the lungs. So we'll move from the blood vessels into the lungs for ultimately going out of the lungs. So let's start from inhalation. Let's start from atmospheric air. In atmospheric air, you have the partial pressure of oxygen as 159 mmHg and carbon dioxide is very, very less. And talking about alveoli, which alveoli is basically the, so let me show you alveoli here. Alveoli are basically here, right? Here, the one that is actually taking place, taking care of exchange of gases, right? So in your alveoli, your air has partial pressure of oxygen is 100 and partial pressure of carbon dioxide is 40 mmHg. Now, just compare between alveoli and atmospheric air. Now, because your partial pressure of oxygen or PO2 is lower in your it is higher in your atmosphere but lower inside the alveoli, what happens is the oxygen diffuses like it can move inside this thing, right? Air. Right. Now, this is about atmospheric and alveoli. Now, coming to alveoli and the pulmonary blood, which is basically bringing the blood, right? Now, you have in the lungs, when the blood enters, the PO2, the oxygen pressure is 40 mmHg, and your PCO2 is your 45 mmHgs, right? And the blood that leaves the oxygen has PO2 100 mmHg and PCO2 40 mmHg. So what happened here is your oxygen diffusing out, diffuse down its partial pressure gradient from the alveoli to the blood. First, the oxygen moved from air to alveoli. Now it has moved from alveoli to blood. And carbon dioxide, again down its gradient, moved from blood to alveoli. And then later from alveoli, it moves out to the atmosphere. Right? Now, this is at the pulmonary scale. Now, at systemic level, again, system is basically your blood is being circulated. Your PO2 is maintained at 9500 mmHg. This because oxygen is bound to the hemoglobin, and your PCO2 is moving as bicarbonate, so it is 40 mmHg only, whatever it was exiting lungs as. Now, at tissue level, so this is a tissue, okay? Now, at tissue, you have the partial pressure is 40 mmHg, and your partial pressure of carbon dioxide is your 45 mmHg. Now, what happens here is oxygen will diffuse from the blood into the tissue, and carbon dioxide will diffuse from the cell into the blood. Okay, all of this movement is based on their partial pressures. Okay, so ultimately, when your blood returns to the heart, right, when it goes back to the heart, it is PCO2 is 40 mmHg and PCO2 is 45 mmHg, and with this only it enters into the lungs. Okay, so everywhere we already know the movement of your oxygen and carbon dioxide through your blood vessels and your tracts. You just need to remember with terms of it partial pressure. Okay, and that basically covers your respiratory tract and respiratory system. You need to be careful, however, about your partial pressures. Okay. Another topic, because we are talking about blood vessels here, we also spoke about your arteries and veins, the different kind of arteries and the different kinds of veins that you have. So I told the first difference between arteries and veins in general is the way they carry the blood. So artery carries it away from the heart, veins carry it towards the heart. um, your arteries basically carry oxygenated blood, the veins carry the deoxygenated blood. Your arteries, remember this point, arteries are under high pressure, veins are under low pressure. Why? We have already discussed. Arteries, because it is high pressure, it is also thick and muscular and elastic. Veins don't really have to be thick, they are thin. And arteries are narrow because of the coverings, and that's why it's under high pressure. Veins are wide in nature, right? And in veins you have valves, so you don't have backflow of blood here. In arteries it's already narrow. Okay, right. Now, along with this, you need to remember these different layers that are there, right? So for example, in your arteries you have your as you can see here, you have your tunica intima. Tunica intima has elastic layer and endothelium. Then you have tunica media and tunica externa. You have the very same layers in veins as well. Okay. Now, if you talk about elasticity, we already spoke. Arteries are more elastic because arteries have to stretch during diastole and they have to come back to its normal position during diastole. So in systole, they have to stretch and contract during diastole because they are under high pressure. Veins don't have to do that. Veins really don't need to do that. Uh, Thickness, we already discussed that arteries are more thick because they have such high pressure, they need to sustain it. Veins are less, experiencing less pressure. Okay. And you have these finer types of veins that are there. So you have large vein, medium sized vein, venule, and then you have fenestrated capillary, which are basically endothelial cells which are have perforations in between. While for arteries you have elastic artery, you have arteries in muscles, the muscular artery, then you have the smaller arteries which are arteriole, and then you have a capillary which is very fine endothelial continuous endothelial covering. Okay, that you need to remember these differences as well. And associated to this was atherosclerosis. If you remember, we discussed about atherosclerosis as the deposition of plaque, which basically means that your arteries are already under high pressure, and because of this deposition your lumen becomes even narrower and the pressure increases, and this leads to something called high blood pressure or hypertension, right? So that's atherosclerosis, and that's a problem because of the deposition of cholesterol, and that's why you are at the risk when you have cholesterol rich food for high blood pressure, right? And hypertensions. Okay, now this we did uh, in the last session also, which is basically talking about your blood flow uh, in your different kind of blood vessels based on the different parameters like vessel diameter, how does that influence your blood flow? Cross sectional area, how does that influence your blood flow? Average blood pressure and your velocity of blood flow, how does that change across blood vessels? So I will not go into very detail because of course we have discussed this already, but just quickly to mention, your vessel diameter is basically showing that for the elastic arteries, they have the largest diameter, of course, because they can open up. And as the diameter rapidly decrease, as you can see, uh, the decrease as your vessel becomes smaller and smaller. And you can see that as your vessels decrease, your blood flow also. So basically, these are talking about different parameters of all of your blood flow. And if you talk about blood flow in this relation, you will see that we have discussed this, that blood flow is inversely proportional to your cross-sectional areas. Blood flow is also dependent on your resistance. Blood flow is dependent on your length. So all of these different parameters are summarized here and here. So we are basically showing you that if you have more resistance, you have less blood flow. So anything which positively affects resistance will inversely affect the blood flow, is what we are trying to say. For example, viscosity is directly proportional to resistance. So if you have more viscous, if you have more cells in your blood, then your resistance, the flow of blood will be less because there will be more uh, factors impeding the flow of blood. Similarly, if you have vessels length very high, that has very high resistance, your blood flow is less because there is high chances of friction. But if your blood vessel radius is your small, or blood vessels is inversely affecting the resistance. For example, your vessel radius is big, so your resistance is very low because you have more blood flow that can easily pass as compared to smaller blood vessel which can give more pressure and the blood flow will be slower. So these two slides are talking about how blood flows is regulated by different parameters, right? And the last this thing lecture of this course covers about different systems. So of course I will not go into very much detail about these things here. This will be of course discussed extensively in the Monday session. So we session 12 will be covering all this topic in great detail, but I will just quickly tell you what do these different systems do. So first of all is endocrine system. These are basically those systems which have glands that secrete hormones and they directly release them into bloodstream, and these hormones regulate several activities in our day-to-day life. For example, metabolism, growth, reproduction, hemostasis, and so many others. And you have several glands, for example, pituitary, thyroid, parathyroid, adrenal, pancreas, the gonads, and the pineal glands. And all of these have their specific hormones which have very specific function. For example, let me just give you one. You have thyroid, which releases T3, T4 calcitonin hormone, and they are responsible for your metabolism and calcium regulation. Similarly, you would all know about flight and fight response, which comes from the adrenaline and non-adrenaline, which comes from the adrenal gland. And this is usually in normal scenario called adrenaline rush. So the word comes from the hormones that is released, right? So you have all these different glands that release different hormones into the bloodstream. And then you have this HPA axis, which is basically telling you how this entire regulation of release of hormone is taking place. So you have a feedback, which is basically, let's say, your body says that I am in fears or I need to do something very quickly. So that feedback is received by the hypothalamus, that is part of this H. Then it releases corticotropic releasing hormone, something called releasing hormone, which will direct the gland to release the hormone. So it will release a releasing hormone that will, let's say, go to the anterior pituitary, which is the P here, pituitary, and that will release the releasing. So this is the releasing hormone. Then they have the hormone which releases the actual hormone from the adrenal cortex. So HPA via these intermediary hormones, and ultimately you have the cortisol, which will make you act fast and respond quickly. So HPA axis is what acts and ultimately leads to the release of hormones from the adrenal, especially in situation of flight and fight. Okay. So all of these are the different hormones that I was just talking about, and each of them have a very targeted approach. They have a very specific targets where they bind by receptors. And of course, if you have problems in these hormones, if they are too high or too less, you have their associated problem. For example, too much of thyroid related problems is hyperthyroid, too less thyroid is hypothyroid. So all of these and several others are determined by the level of hormones that you have. And like I said, you have adrenal glands, which basically covers how much of your like how much each of. So first of all, your adrenal gland in itself has several regions that can release different kind of hormones. So you can imagine the level of regulation your adrenal will have, and that's why that HPA axis becomes very important. Okay. Next system is digestive system, which basically is all about how you eat and how you digest your food. So you have all these, the main components of digestive systems are mouth, esophagus. So you eat, then your food pipe takes it to the stomach. Stomach does the initial protein digestion and acid secretion that takes it to small intestine, where you have the absorption of nutrients. You have liver for metabolism. Pancreas releases the enzymes which are responsible for all these digestions. And lastly, you have large intestine for absorption of water, and then you have the excretion process, right? So that's how your digestive system. But what is important is at each of these steps that I just told, there is many hormones that are released and many enzymes that are responsible. So we will be discussing that, but it's a big topic, and it will never end if we start discussing about it. But you need to understand it in the main. So the main components are written here, and you need to remember it in a flow chart way so that you can connect the flows. Okay. Okay, the next topic is about blood. We spoke about everything about blood. We didn't discuss about the blood itself. We spoke about the vessels, but not the blood. So the blood carries three parts: plasma, buffy coat, and the red blood cells, which is making up the major components. And in the buffy coat, you have the white blood cells and the platelets. And the plasma, you have the salt and the water and the proteins. Okay. And formation of blood cells, all these different cells is called hematopoiesis. And you can see that there is one major stem cell, which is hematocytoplast, which gives rise to these different kind of cells of blood. Okay. Then we spoke about absorption. So it becomes very important to understand kidney, which is important for filtering of blood, for all the absorption of, like electrolytes, acid balance, base balance, urine, so excretory system, blood pressure. Kidney is a very important system. And with relation, so that's what I was saying, that they are responsible for metabolic waste removal, fluid balance, blood pressure. All of these are important functions of kidney. And lastly, you have the RAS, which is basically the renin, angiotensin, aldosterone system. This is basically your kidney cells, how they respond to renin. What is renin? Renin is released in response to a lower blood pressure. So when you have low blood pressure, your renin is released and released by the juxtaglomerular cells in the kidney. What they do is renin converts something called angiotensinogen into angiotensin 1. Angiotensin is converted, 1 is converted to 2 by ACE. And what does this angiotensin 2 ultimately do? It does vasoconstriction, and it increases the concentration of aldosterone. What does aldosterone do otherwise? It releases, it leads to increased sodium and water retention. So what does it do? It increases the blood pressure. So what is basically this? This is the way of hemostasis, your regulation. Remember the first thing that we discussed about. So this is a cascade of how your body regulates your system, right? Okay. And then the next system is immune system, for which there are, of course, as you know, several things that you need to remember. But broadly, you should remember the what is innate immunity, something that you are born with. So the physical barriers, phagocytes, complement system, this is innate immunity. And then you have adaptive immunity, which you develop over your lifetime, which comprises of B cells, which is all the antibody, and the T cells, which are the cytotoxic and the helper T cells. And you need to remember the major lymphoid organs, bone marrow, thymus, spleen, which are all part of your immune system and act together for defense against pathogens, toxins, and any abnormal cells for their removal. And immune system, one very important thing that you should remember is, one thing is immune system is specific. Second thing is immune system has memory. B cells are making antibodies and they're also memory. So what is memory? Memory is, let's say you have a virus which has infected you right now, or let's say a bacteria, and you have made an antibody. Next time if that pathogen affects you, you deploy the same antibody. So your immune system also has memory. And along with that, it has self-tolerance. You will never, and the perturbation of self-tolerance is autoimmune disorder, where your body itself thinks that, okay, my own cells is a problem, so I should attack it. So that's autoimmune disorder. But otherwise, it has something called self-tolerance, where it knows to differentiate between self and non-self. And whatever is non-self is foreign body, and I should act against it. And whatever is mine, I should not act against it. So very important system for, or very important characteristic of immune system. Okay. And I think I have reached almost towards the end, which is the last system that we are dealing with this in this course, this reproductive system, which talks everything about how this the system of female and male produce gametes, sex hormones, and how is the entire procedure of reproduction supported. So we will see that in males, we will read about the organs like testes, epididymis, and then in hormones, for hormones, we will see that in male system, you have testosterone, FSH and LH, all with their very categoric functions which affect sperm production, how they are delivered in the reproductive tract of the females, and how are they, what is governing their features, what is the structure of sperm, how are they formed. So it's a very again very controlled procedure. Similar to females, we will see that the organs which are ovaries, fallopian tubes, uterus, the hormones are estrogen, progesterone, FSH, LH, and the functions of the female reproductive system basically has to do with egg production. So in relation to that, you would have seen about menstrual cycle. Then you have fertilization, and after fertilization, when you have the zygote formation, how does the body uh, makes itself ready for the childbirth and the pregnancy. So everything. So this is just one of the ways of which shows you the fertilization. So we'll discuss this in the problem solving session 12, and there we will look into this in more details. Okay, so we've already covered more than 2 hours, I guess. So what I will do is I will not discuss this question. Just wait on this slide and give you the answer for your perusal. The false statements correction will be given to you in the PPT, which I will be uploading. So all of these are there, and you can refer there. So you can answer and then look into the this thing options here. I'm sorry, some of these already have the answer, but that's okay. You can just read and check the answers, right? So the answers to the fill in the blanks are also there. So what I will do is when I give you the PPT, I will correct the animations and give it to you. Similarly, you have these as a fill in the blanks, and you have a lot of match the followings, right? So with that, we come to an end of this session, and all the best to everyone who's attempting the exams. And I hope you enjoyed the course and the problem solving sessions, and it was a nice time and nice experience to share the information with you. Thank you very much.